So I'm very happy. This is the closing session of the sessions of the conference. It's not there are several sessions, but this is the last session. There will be lunch after this, okay? So if you're hungry on that, so just let you know. And I'm very happy to have Dr. Ilana Kolodkin-Gal give her first talk. Thank you very much.
Shalom lekulam. Can you hear me? Okay. I got additional 5 minutes from the chair, so I would be on time, but it would be 15 minutes. And I would like to speak about below-ground interactions from my perspective. So what I try to do every time is to understand how all the different things which are circled in green are occurring molecularly, which means how do bacteria interact with different partners underground. In order to do that, I use a very useful working horse, which is beneficial bacterium associated with the root, which is very well characterized for gene expression. For example, you can see here in red cells that stick to the root, in blue the spores, which we coat the seeds with, and in green the cells that would disperse between roots in one colony. And we can really understand why bacteria are converting from one situation from another by studying how they express a given gene set in a given time. And here you can see the ones that stick give rise to daughter cells that swim. And the question is, how can we take all this information into the environment? Something that we did routinely in the last years. Now, it's not just a philosophical question. It's a highly practical question because the same bacterium is already a product. So we add Bacillus subtilis and its relative to plants to protect them from fungal and bacterial pathogens. And we do it all the time. And in order to understand it efficiently, we need to understand how bacteria interact with the plant. So very early on in 2019, we made the notion that the root actually selects for the most beneficial colonizers. And the reason we made this claim is because we competed a series of beneficial bacteria to the plant. And the most beneficial bacterium, in this case Bacillus subtilis, always won in the competition, but only if the plant was present. Which actually tells us that the plant in this interaction is not naive. And it does more than providing to the bacteria carbon or nitrogen. It actually changes how they interact with their environment. Sorry. Okay. Okay. I hope you can hear me now, okay? So how can the plant select for beneficial colonizers? It can give them carbon, nitrogen, and trace nutrients. It was shown again and again for growth. It can induce their stress tolerance. We know that bacteria improve the stress tolerance of their plant, but this could work the opposite direction. The plant could increase the stress tolerance of the bacteria. It could give them advantage during competition, for example, by inducing specific antifungal and antibacterial compounds. And we need to remember it can have short-term and long-term effects. Short-term effects as long as I'm associated with the root or immediately after I detach. Long-term effects, if I detach bacterium from the root for generations and generations, it would express genes which are associated by the root. It would have a memory of once being associated with the root. So in order to know if this is the case, we did a very simple experiment. We took Bacillus, grew it with Arabidopsis, detached it, and did transcriptome. This is physical and chemical interactions with the root. We exposed the bacteria to the secretions and did transcriptome. This is just the chemical interactions with the root. And then we moved bacteria to a fresh rich medium and grew them for 8 generations and did transcriptome. And now we get just the genes which are memorized by the bacteria. Genes which were induced by the root and maintained over generations and generations in the bacteria. And we have all the appropriate controls, of course. So the first thing I want to say that chemical interactions with the root in this set is very similar to physical and chemical combined. So most of the signals are chemical, and we know that because the effect of secretion is very, very similar to the effect of the plant. The main thing that distinguish the two groups are metabolic, and I will not go into details here. Bacteria which grow without secretions are clustered completely elsewhere. Now, do bacteria remember being associated with the root? And the answer is, surprisingly, yes. When we grow them for 8 generations in LB, we lose most of the signature of a gene expressed in association with the root. But there are 278 genes which are really significantly induced, even 8 generations after you grow the bacteria with LB. We neutralized all artifacts that come out from the media, and although we do not see it in PCA, like many things which are not seen in the PCA, we see it very specifically when we look gene by gene. So bacteria also remember being associated with the root. Now, what genes do we see? So we see a lot of genes, especially in the shorted response, which are associated with metabolism. We see genes that allow the bacteria to stick to the root, like biofilm genes. But we see, and this is a new group in this type of context, we see the root is inducing robustly genes which are associated with stress tolerance in its beneficial bacteria. And you can see it in here in red, okay? And we can see together with that a pathway, cyclic DMP, which is tightly associated with stress tolerance in bacteria. And we also see induction of antibiotic production genes, and in simplification, we would say the bacteria are inducing the plant's systemic immune response, but the plant is inducing the bacterial immune response, which is producing antibiotics to protect them from fungal and bacterial agent. And as we go to the long-term response, which means 8 generations after growth in LB, we lose most of the signature of genes which are induced by the plant, but we are remained with genes which are primarily associated with stress tolerance. So does it have any phenotype, right? This is just expression. Expression means nothing. So let's look how bacteria are growing in the presence and absence of stress. So with secretions, we see tiny effect on cell growth. But when we add abiotic stressors, which is salt, which is highly relevant to arid environments, we see dramatic difference. Bacteria do much better in the presence of abiotic stress if they were exposed to plant secretions. And we can quantify the lag phase and see it again. The lag phase gets much shorter. This is abiotic stress. What would happen with biotic stressor? And we use salicylic acid because it's highly relevant to root-associated bacteria. It's a part of the plant immune response. And in fact, bacteria are killed by high concentration of salicylic acid unless they were exposed to the root first. So if they were exposed to root secretions, they become fully tolerant to salicylic acid. Do bacteria that remember the root are also more stress tolerant, as the transcriptome predicts? And the answer is yes. So again, this is already in the descendants. From the 287 genes, 60 are stress tolerance. And we can see that the D genes are still induced in the descendants. The effect on growth almost completely disappeared, but the bacteria are still much more resistant to stress. This is growth in high salt with no secretions, again, in the far past before they met LB. And this is with salt if they once met secretions of the root or the root itself. They also compete better on the root, as you expect from the fact they are more tolerant to salicylic acid. So if we take bacteria that once met the root and tag them with a fluorophore, they would always compete, I hope you can see it in green, better for root colonization than bacteria that never met a root before. And this is independent of the fluorophore. If I'll switch to the fluorophore, I'll get exactly the same behavior. So they are also more fit in a competition. Once interacting with the root, you become more fit for a very long time because we see the same effect in the descendants. And I will just remind you again, we do see a transcriptome different in the descendants as well, which means we have a memory. We also know which genes induces the memory. I will not speak on it today. And the last thing, and I just want to remind everyone, I'm fully aware to the fact that Arabidopsis is not a crop, and it's also not that important, which is really sad. Please do not cite me on that. So we also did it with tomato. So this bacterium is very beneficial to tomato. It promotes its growth and protects it from fungi. And when we do it with tomato, we get the same effect. The signals from Arabidopsis induce a competitive advantage over tomato. I would just say that the signals from tomato are not that good. So it's specific to a given host. Whatever Arabidopsis is producing is really important here. I would just make the point. It is well known that bacteria induce the plant systemic immune system. And we can look at antibiotics as the systemic immune system of the bacterium. This is how it protects itself from the hostile world. And when we look at the transcriptome, it's very, very clear that the plant are inducing antibiotic production. They are not naive to that. They are really good at that. And when we look at the ancestors and also in the descendants, which are introduced to the plant, we see repeatedly induction of antibiotic production genes. Now, I would not make this point, but if you want to know more on this particular antibiotic, which is induced by the root, you can read our Nature Communications paper from this year demonstrating how this antibiotic is most important to the interaction with 11 other soil bacteria. So, I skip that. Do we have a signal from the root? Obviously we do. Otherwise, I wouldn't have been here. We tried a library of hundreds of molecules from the plant with Aasfar Abderrahim. None of them worked. That sucked. And they didn't work for growth or for the expression of genes which are associated with stress tolerance. But, and this would be the light point, we tried a library of Arabidopsis mutant. And as we did that, we could identify a mutant, and here we look on stress tolerance genes, in the DXR enzyme, which loses the signal altogether, which means all the mutants in DXR are basically comparable of having no secretion. So we extract the potential signal from these mutants, getting absolutely nothing. So what I told you is that bacteria interact with the root in a very interesting way, having nothing to do with metabolism, although metabolism is important, and that it not only the bacteria protect the root protects bacteria back, and this interaction has a memory. What I didn't have time to tell you is that we did the genetics of everything that could be related, and we have the specific stress response which we acting downstream to the plant in bacillus, and also what activates antibiotic production and how they interact with each other. And if you want more, please read our bioarchive paper. And the students that did this work are Harish and Omri. It is now continued by Ronit. And we are really, really interested to take it into synthetic communities, which would, when I'm saying synthetic, I mean a signal and a bacterium together, which would protect the root better, and also to understand better how feedback from competing members of the community in the rhizosphere affect the interactions and the signaling pathway, which I just described. So thank you very much for listening.
So thank you very much. So we have time for questions, or yeah, Ariel.
Maybe you did mention it, but to what extent the memory effects that you find are a result of simple Darwinian natural selection or versus epigenetic?
So the first thing we want when I say epigenetic, I need to rule out evolution or selection for a specific genetic trait. We sequence the genome enough to know there are no mutations that explain the phenotype. And we also did an experiment where we grow bacteria with secretion. We take 100 isolates and regrow them. And nothing appeared as different if we go through the LB plating and isolation. So we have every reason to believe that it's not genetic. It disappears within 12 to 15 generations. So it has a duration. And you can explain it by cycles of cyclic DMP biosynthesis. The plant is actually inducing adenylyl cyclase, which induces stress tolerance, which induces another adenylyl cyclase. And between the two of them, you have enough molecules of cyclic DMP in the bacterium to grant some memory of being associated with the plant.
So any other? One more question or comment? So I would like really to thank Ilana for her great talk and for being on time, and really thank you very much. Our next speaker is Dr. Omri Finkel from the Hebrew University, who is first time here in our campus, so I hope you treat him well. Thank you very much.
Okay. So, yeah, let's dive into it, right? So when we talk about microbiomes, this is a relatively modern word. There's some argument about what its etymology is. What does it mean? Some people say, well, microbiome is microbial biome. It's a hilchem. Other people say, no, a microbiome is analogous to a genome, right? It's another ome, which implies that a microbiome is basically an adaptive trait of a macroorganism, right? So, and we can try to find parallels between microbiomes of people and perhaps microbiomes of plants, and then ask ourselves, well, did the plants actually evolve to select particular microbes which actually help them in the environment, and how does this evolution occur? Especially when we consider that we know most of these microbes, most of the bacteria and fungi that associate with plants are not maternally inherited. They are recruited from the environment every generation, every new generation. So going into my postdoc a few years ago, I was asking, oh, well, before that, I'll maybe show this. So in some cases, it's very easy to show this kind of stuff, where we see a very strong adaptive signal, right? It sometimes made me a little bit envious that people working with nitrogen fixers can, nodule-forming bacteria can get these amazing phenotypic effects when they add. So then, recently in my lab, we decided to get into this too. So this is work from Moshe Alon from my lab, where this plant is Calicotoma villosa, Kida seira, growing in a soil that doesn't have any of its symbiont. He adds a symbiont to the soil, and voila, the plant is very happy. But in a lot of cases, the signal isn't as clear, right? We have not just these cases of very, very distinct symbioses, but more maybe subtle interactions between the plant and the microbiome. And what we ask ourselves is, how do we tease these apart? How do we dissect these different interactions when we have thousands of different microbes, and try to understand what's going on, right? So when we know that all of these microbes, not only in nitrogen-fixing plants, are definitely useful for the plant. This is another experiment we did in the lab that demonstrates this quite simply, right? So we have plants, Arabidopsis this time, so not nitrogen fixers or anything like that, growing in regular potting soil. Microbiome's intact, plants are doing fine. We destroy the microbiome in the soil. In this case, we do this by gamma irradiation of the soil, and we plant Arabidopsis in that, and they're miserable. We don't really know why they're miserable. This could be for various different reasons, but this process of destroying the microbiome is not good for the plants. But we can then add a synthetic community. We can add a collection of microbes we have growing in our lab, just not necessarily coming from the soil. And this completely recovers the plant phenotype, right? So this demonstrates that the plant definitely needs these bacteria. But this isn't always the case, right? So when we grow plants in agar, as I did a lot during my postdoc, right? And these are conditions that the plants absolutely love. They have very easy access to all the nutrients they need. And we grow these on sterile agar, and the plants are very, very happy, right? We add bacteria, and if anything, we get a bit of a little bit of generally larger shoots, but shorter roots. So the plants don't intrinsically need these bacteria. These are used in environmental contexts. So yeah, so going into this postdoc, I was doing. These are the questions I was asking myself, and we figured, well, there's hundreds of different bacteria, thousands of different bacteria in the soil. How do we come about addressing these questions when we look at one of these bacteria at a time? Doing that, we first of all will, die of old age before we look at all the different bacteria, and secondly, we're not considering all the different interactions between the different microbes. So we said, well, why don't we emulate a natural community by building what we call synthetic communities, do this in a microcosm, when this microcosm encompasses all the different interactions, everything that's happening, but it's also very, very highly tractable. We can start playing around with it, changing environmental variables one at a time, pulling some stuff out, putting some stuff back in, but we have full control over the system. So this is what we did. We used this synthetic community. It's based on a collection of 200 bacteria that were isolated from Arabidopsis roots. And let me just pull this out. Just standing still here. Yeah, so we build this community that's isolated from Arabidopsis roots, and we inoculate the plant with these communities in a gnotobiotic system where there's no other microbes, just these communities. And just based on this system, this was about a couple years of work where we manipulated a lot of different environmental variables. I'm not going to walk you through that today. I'll just show you sort of the highlight of this was where we realized that there's one particular group of microbes that was always, in all these cases where it was absent, we had a very strong phenotypic effect on the plant. So this is not a case where we add a bacteria and we see something, we remove something from a full intact community, and we see a very strong effect, right? And we demonstrate this in this experiment over here. We call it a dropout experiment, where we have a full community of almost 200 microbes. And then we compare it to the same community, just removing the This one gene is called varivorax, and we have a very strong effect specifically on the root growth. So basically, the roots don't grow at all. This is the length of the roots at the beginning of the experiment, because we start with 10-day-old seedlings, right? And that they basically just don't grow. So we can show that there are particular microbial groups, in this case varivorax, that are absolutely necessary and sufficient in the context of a full community to maintain plant growth, right? Considering all the different microbe interactions. Now, the thing is about these varivorax, that just on their own, if you inoculate a plant with varivorax in sterile conditions, you see nothing. There's no effect. We'd assume that they're absolutely inert with respect to the plant. So we got to assume that this is a microbe interaction, and indeed this is the case. But it's not a type of interaction where there's any kind of antagonism between varivorax and any other microbes. It's actually a metabolic interaction where varivorax is very good at inactivating auxin. So it's a very, very good auxin degrader. We can show this with plants that are auxin bioreporters. So this is a plant, Arabidopsis plant, that have GFP fused to DR5, which is a synthetic auxin reporter promoter. Right, so if we grow the plant without bacteria, this is the root. We can see a bunch of GFP here around the meristem where the endogenous auxin is produced. If you add auxin-producing bacteria from the community, which have very strong effects on root growth, you can see that the root turns green. You see some green all over the root. But then if you add one of our varivorax, in this case, this is a strain we call CL14, we completely lose the signal. So these varivorax are very good at degrading auxin around the root and diminishing this very strong signal. So how do they do this? Well, that's another advantage of using these synthetic communities, right? Because they all have sequenced genomes. And we can compare the genome. So we ask ourselves, well, what are the genes that all of the varivorax have? Because all of our varivorax were able to do this, and none of the other bacteria in our community have, right? And then we can have this just a list of genes. We see them down here. All this black basically signifies a bunch of different genes in different columns that are unique to varivorax. They're also grouped by proximity on the chromosome. So we have these different we call them hotspots of varivorax unique genes. And when we do a transcriptome for varivorax, in the presence or absence of these auxin-producing bacteria, we can see that some of them are strongly induced, in particular this one hotspot we called hotspot 33, where you see it's all turning red because it's highly enriched when auxin-producing bacteria are around, right? In parallel, we can also take we also took a more functional genomic approach where we used the fact that auxin is very easy to measure using a colorimetric method. So we made an E. coli library a phosmid library with varivorax genome in E. coli, and we looked for E. coli clones that have this phenotype, right, where they're able to degrade auxin. It shows up as the media turning white. Right, so we scanned this entire E. coli library it's about 5,000 clones for auxin degradation, and some of you may have noticed already. We actually found a couple of these that the media does turn white. So these are auxin degrading E. coli now. When we sequenced the varivorax DNA that came into this E. coli, the varivorax genomic region, we found that these both have two different inserts, but they have this overlapping region, which happened to be hotspot 33. So we found this one, basically operon in varivorax, which appears to be implicated in auxin degradation. And indeed, when we knock out this operon from varivorax, we lose the phenotype. So it's no longer able to, this is our knockout strain, it's no longer able to rescue plants from this short root phenotype that's induced by auxin-producing bacteria. This is what this operon looks like. Some follow-up work in my postdoc lab, Jeff Dangle's lab, here basically got the structure of the transcriptional regulator, a MAR regulator found in this on this operon. You can see the auxin. It's a dimer. Auxin is binding here. When it senses auxin, repression is released, and this operon And this operon turns on and starts degradation in Varivorax. Another group in China just recently, saw this in bioRxiv, they took the same operon, they saw our paper, and they managed to get a cryo-M structure of the actual enzymes in the operon that are degrading the auxin. So now we know what's actually happening to this to the auxin. It turns into what they call OX-IAA, which is basically an inactivated variant of auxin. So it's really nice always to see people taking your work and taking it to new places. That's always cool. Yeah. So and finally, just another. This is some more follow-up work at Jeff's lab, right? So we know auxin degradation isn't. We didn't discover auxin degradation. There's lots of different bacteria that can do auxin degradation. So we were very keen on showing that our auxin degrader is the best and you know better than other sort of canonical auxin degraders. So here we have this competition happening where we substitute Varivorax with different bacteria that have the more canonical auxin degradation operons, and you can see here we have it here. It's basically enough to just add one Varivorax strain to these large 200-member communities to rescue plant phenotype. So we don't need all 10 in the first cases. We have just one, and none of the other strains are able to do this. So yeah, Varivorax are definitely special. Yeah. Another follow-up from this work, which was kind of fun. This came from Becky Bart's lab in Danforth Center. They have this cool phenotyping system where they look at sorghum, and it's a desert, drought, and drylands conference, so this is under drought. And they asked us to send them bacteria, to send them synthetic communities to test on the plant, but they told us, don't tell us what's in them, don't keep this experiment completely blinded. So we tricked them. So we said, okay, we have Syncom A, Syncom B, and Syncom C. But what we didn't tell them was that Syncom A was basically all the bacteria in our collection that we knew are kind of good guys that didn't harm the plant. Syncom B were all of the bad guys, all of the really harmful bacteria that we knew in our collection were these guys. And then Syncom C was actually Syncom B plus Varivorax. So in the paper, we changed the name, but because they already knew the answer. So it's basically exactly this Syncom just with Varivorax, and what you may be able to see here, that's what the Syncom produced in the plant, the Syncom B, the bad guys, right? So under drought, not so much under well-watered conditions, but under drought, this bad Syncom definitely harms or negatively affects plant growth. Syncom A, the good guys, are fine, but then when you inoculate it with the Syncom B plus V, so when you add only Varivorax to the bad guy Syncom, right, you have a complete recovery. So this is plant growth promoting per se. We have the same growth as we do with the control, but we know which bacteria in a natural soil community might be important for plants to develop properly and produce biomass. How am I doing with time? Oh, perfect. All right, so it's just enough time to tell you about. So this was all postdoc stuff. I have a little bit of time to tell you a little bit about what we're doing in our lab. So I recently opened a lab at Hebrew University in Jerusalem, and we're trying to do a couple of things along the same line, which is to try to understand how bacteria affect plant phenotypes within this full context of an intact community. So we came up with this strategy. We call it a top-down dropout approach, where we have a notobiotic system where we grow plants in the presence of a full community of bacteria. So, same 200-member community, or maybe another community we isolate from different places. But then we start dropping off sections, whole large sections of this community. In this case, this is just guided by taxonomy, but we can use other features to guide this type of dropout. And then we look for a phenotype, right? We look for places where the plant is unhappy, right? And then just like we did before, we assume that this unhappy plant is now unhappy because we're not including this whole group of bacteria. Then we can do other iterations where we drop out smaller and smaller subsections of this group of bacteria and ask ourselves, again, within a context of all the different interactions, which are the important taxa, and eventually, hopefully end up with this one focal taxon. So to start off with this, a very talented master's student in my lab, Amal Mahalwis, is doing this exact same experiment where the phenotype we're looking at is basically blocking a bacterial pathogen from invading the leaves. This is kind of weird because we're using soil bacteria to look at a leaf pathogen. This is Pseudomonas syringae, DC3000. But it turns out there we do see very, very different effects. And it's easy to measure because this is a luminescently tagged DC3000, so we can just count its colonies in response to different bacteria. And it turns out, so here's this basically this setup. It's a bit messy, but this is the setup of the experiment here. This is the amount of pathogen we're able to recover from the leaves, and we have, either dropping out each one of these different phyla, actinobacteria, Bacteroidetes, firmicutes, or proteobacteria, or just putting each one of these phyla alone. And again, this is the best looking figure, but you may have noticed that when we have only A or only B, actinobacteria or Bacteroidetes, so the plant is fine, but when we have a lot of pathogen, right? But when we have only F and only P, so only firmicutes, bacilli, for instance, and only proteobacteria, these two groups are sufficient to significantly reduce the amount of pathogen on the leaves, and we see the exact mirror image when we do the dropout. So when we pull out either the proteobacteria or the firmicutes, we have a lot more pathogen on the leaves, right? So our next step is to do the next iterations and find which one of the taxa within these groups in a community are having this effect. Another project we're just getting started on in the lab is trying to, again, go back to this question of adaptation, and we're taking a step back from synthetic communities now, and we really want to start looking at natural communities and understand whether plants in the desert are more adapted to deal with abiotic stresses related with the desert than plants that don't grow in the desert, right? So we have this, we have folks around here working on this kind of stuff, and we have a lot of ecological information of different plants, and I'm showing here Brassicaceae. So we can couple very closely related plants that are either typical of northern or southern areas in the country, and conduct comparative experiments where we try to identify microbes that are unique to desert-adapted plant species. Very excited to get this project going, right? So, and what we're really after is these genes that in desert plants under drought stress, these bacterial genes that are under drought stress in desert plants will be induced. Right, so that'll be it. I have a lot of people to thank here, many people in the Dangle lab that contributed a lot to this work, and the different collaborators. These are some of the members of our group, and thanks to them, starting to do some interesting work, and thank you all very much.
So thank you very much, Omri. So any questions, comments? Yes, Ilana.
So a beautiful talk, and again, it would be a naive question. So you said the vermivorax need to degrade auxin that comes from auxin-producing bacteria.
Yes.
But the auxin-producing bacteria probably have a very important role, because without bacteria at all, you get no growth, right? So most likely, if you had to design an ecological scenario, it would have to be coupled to the thresholds of auxin in the system, the auxin degrading operon.
Yeah, so you might, right, I kind of glossed over that part, but right, it's auxin, and this, obviously plants need auxin, and auxin in this system has conflicting roles, and there are many papers that show that auxin-producing bacteria are beneficial to the plant. My view is that the reason bacteria in the soil are producing auxin isn't necessarily to help the plant. The plant is perfectly capable of generating its own auxin, and it also needs very, very close control over where the auxin is and where it's going, right? And it has none of that control when bacteria are producing it. But I do think that it definitely has an adaptive value for the bacteria, whether they're good or bad bacteria, quote unquote, for the plant. Right, you'd get a different response. Well, many pathogens produce auxin, but also many beneficial bacteria produce auxin, and I think it has to do with just a way of making the plant more amenable for colonization for these bacteria. So if it's a beneficial bacteria, it'll make the plant more amenable for colonization, and you have a beneficial effect, and vice versa. Vermivorax, of course, probably is just using this compound for its own use. It's food for it. And it's kind of, cleaning up the root. But there's still some, this is a sort of a story that there are still some gaps in it where I'd, be happy to fill.
Yeah, yeah, of course.
Thank you. I have a lot of votes. So oxen, going on the same topic, I think. Oxen is a great signal for the rest of the plant to divert nutrients and carbohydrates towards the organ flaxing oxen from.
Mhm.
So, do you see any allocation effects or even morphological and architectural effects of that under your treatment?
Yes, so
Repeat
should I
the question, because on the video they don't hear,
Yeah.
okay?
Oh, yeah. So just to repeat the question, so I was asked, auxin is an important signal to allocate nutrients and resources to particular parts of the root. And the question was, do I see morphological effects or different kinds of effects that are sort of spatial and found in different parts of the root in response to the bacteria? So yeah, the answer is yes. So we see, right, so both if we add exogenous auxin to a system, or if we add bacteria, we see similar, auxin-producing bacteria, we see very similar effects. So, right, so we see that the root architecture completely changes. Maybe one difference is that, if you add auxin to a system, it eventually is consumed, but if you add bacteria, they're continuously producing it. So we see very aberrant morphologies in the root. We see sort of the root tips are getting sort of inflated, and they stop growing, a lot of different changes. In this particular system, we don't really have a lot of spatial structure. So what we see when we, for instance, use the bioreporter is that the whole root is green. So we don't see that it's allocated to a particular region. It's just everywhere, which must be very confusing for the plant.
So really thank you very much for a great talk, Omri Finkel. So we have 3 more speakers. So the next speaker is Professor Osnat Gillor from the Belowground Institute for Desert Research at Ben-Gurion University.
Thank you. So thank you, Shimon, and really thank you for this extraordinary conference and for inviting me to talk. No? No? Okay. I'll do my best.
Sorry.
Okay. So I'm going to shift gear a little bit and basically go back to talking about agricultural soils. And in the previous session, we talked about treated wastewater irrigation. I'm going to take a different route, a different way of looking at it. I'm more interested in trophic interactions in the soil. And I'm going to just go briefly and tell you that in Israel, as Eddie was saying previously, we are using treated wastewater for irrigation, and we are irrigating plants with treated wastewater. And we had a project where we were interested in what our contaminants are found in the treated wastewater, and how are these contaminants affecting the water, the continuum of water, soil, and crops. So we're interested in the bacteria that are pathogenic, but not only in bacteria, but also in viruses, protozoa, and antibiotic. And I'm not going to talk too much about what we have found. Suffice to say that you'll see that we found very little. And we were trying to understand why that is. So how did we go about it? So we were trying to do everything we could think about. So we were trying to use different types of treated wastewater, so really low-quality wastewater to high-quality wastewater, and fresh water. So these are all field experiments that were done in the field, and we applied everything. We applied different treated wastewater to the field. We applied, and we also used different crops, so tomatoes, cucumbers, and melons. And we tried the different water types. We tried different soil types for sandy loam, loam, and clay. And we were looking not only at the soil, but also at the crop, and we were trying different soil barriers. So we were looking at different soil barriers like plastic mulch, the subsurface drip irrigation, and so forth, trying to understand what's going on. I'm not going to talk about everything because it's been published, but it's not what we're going to be talking about today, but we were looking both at the crops, we're looking at the crop wash and the crop puree, and we were looking to see what kind of pathogens we're going to find. So let's start just with the water. So we were using different types of water, as I said, and we were looking at the water. This is Ezra. Ezra was doing all these experiments for 2 years. This was part of his PhD. And he was looking at the water, and he found, as expected, in the fresh water or the potable water, he found no pathogens, but in the treated wastewater, he found sorry, not pathogens, but indicators. So tall coliform, fecal coliform, E. coli, Enterococcus, found a lot of them. But when we, and that's, no, I'm not going to talk about that. And then when we looked at the soil, nada. So the freshwater irrigated soil, the treated wastewater irrigated soil showed the same a pattern of, or the same profile of indicators. When we looked at the tomato wash, we've seen the same thing, so the same pattern. So that was interesting. So what happened to all the E. coli we put into the soil? And in this case, the soil had lots of E. coli, and we couldn't find anything. And then we looked at the soil barriers. We've seen the same thing. I'm just going to show you what we saw when we did the different soil types. So we used loamy sand, loam, and clay. They're very different. We used them in the field, in the same field. This is BY, and he was doing these experiments, and this is what he found in the water. So more E. coli, more Enterococcus, more fecal coliform, Pseudomonas aeruginosa, and we did that with cultivation, molecular methods, MPN methods. So any known method, all the EPA methods. So we've done everything we could to find these indicators. But when we looked at the soil, and what I want you to see is that we have here non-irrigated soil, potable water irrigated soils, and treated wastewater irrigated soil, and you compare blue to blue, brown to brown, and so forth, and you can see that there's no difference. So there's no statistically significant difference between the different soil types, regardless of the water quality we used. And that was true for the soil barriers as well. And so our main questions are, where have all the treated wastewater indicators gone? So where are they going? We can't find them in the soil or in the crop. So I'm not showing you the crop, but we can't find them in the crop either. So that's a good question, and we were kind of banging our head thinking about how to answer this question. And we actually started looking at very old papers. And one of the things we were suspecting is maybe there is something either killing or eating these bacteria once they're introduced to the soil. And the question was, how are we going to test that? So we were thinking of very elaborate methods, but then we came up with a very, very simple experiment where we used treated wastewater, our synthetic treated wastewater, and we supplemented the treated wastewater with cyclohexamide that inhibit eukaryotic cells. And what we wanted to see is that if we have the synthetic treated wastewater, we add the E. coli with and without the antibiotic, would we see a difference in the survival? And we collected agricultural soil. This is a pot experiment we did. We actually did it with organic soil and with conventional soil, and we tried everything. I'm not going to show you these results because we're currently analyzing them, but just to show you what happened, this is Vali, and she was doing this experiment. So just like Eddie showed earlier, the x-axis is time, the y-axis is the E. coli concentration, and you can see that over time, the E. coli concentration is decreasing, but if we're using cyclohexamide, as you can see, if we're using cyclohexamide, then the decrease is inhibited. So there's the E. coli survive much better when the protists are inhibited. So that was interesting, and we wanted to follow that up. And so we know that enteric bacteria decreased in treated wastewater irrigated soil unless the protists were inhibited by the antibiotics. So the main question is there a difference between the protists in treated wastewater irrigated soils compared to potable water irrigated soil. And are the bacteria different? And so this is kind of a scheme of what we can see here is the water and the different soil types, and then we have the different bacteria. And to make a long story short, we didn't see much differences between the water quality irrigation, but only between the different soil types. Same goes for the different protists. We were looking at the different protists, different soil types, different irrigation. What ruled, and you can see it here in the looking at the biodiversity, so I want you to see is that if we're looking at the bacteria are clustered together according to the soil type, while the protists are probably more generalistic, or some of them at least are more general, and they were found in the different soil types. So we know that the microbial communities significantly differ between the water qualities, but these differences did not carry, So the microbial communities significantly differ in the different water qualities, but it did not carry to the irrigated soil, and the type of soil ruled the bacterial community and the protist community, and that the soil texture, especially clay, was found to be very important for shaping the bacterial community structure, and that the variation in the bacterial communities did not entail diversification of the protists. So we're thinking that it might be more generalist. So the next thing we wanted to do is to see, okay, let's take it to experiment. Let's do some lab experiments, and so we need to isolate protists for that from the different soils. So isolating protists is very laborious. You need lots and lots of patience, and it's not as easy as isolating bacteria. So we were collaborating with June from Nagoya University, and he was isolating the protists, so he was helping us with this study, and he was isolating different protists from the different soil types. These are all free-living amoeba, so we were only looking at free-living amoeba. There are other protists, ciliates, flagellates that are predating on bacteria, so we're not looking at them at this point, but we are planning to do that in the future. And then we wanted to do the following experiment. So taking these isolated free-living amoeba and introducing enteric bacteria and test predation under various conditions. Mainly, we thought about salt, because in treated wastewater we're introducing salt into the soil, and temperature. So we decided these might be very important, and then because we isolated about 13 free-living amoeba, and we were thinking of 3 different bacteria that are known to be enterics, so E. coli, Serratia, and Enterococcus that we found in the environment. We wanted to see how the different temperatures, the different salt conditions affect predation. So this was a huge experiment that a very, very talented postdoc was doing for throughout the corona, so he had nothing else to do. We were quarantined. He can only go to the lab, so he was working day and night. And we had tons of data, and so our collaborators, Nadav Schnerb and Emmanuel, he's a PhD student, both from Bar-Ilan University, were using Lotka-Volterra equations in order to model this, the interaction between the protists and the bacteria, so looking at predation. Basically, these are predation equations. But again, this is their work, and if you have any questions, I suggest you ask them. But what I do understand here is that they model it such that they develop these equations. We're saying that alpha means faster bacterial decline, well, beta corresponds to faster growth of the amoeba. And I just want to tell you that throughout the experiment, Sourav was measuring both. He was measuring the decline of the bacteria and the increase of the amoeba population, and it's not easy. While T is basically measuring the time, and X0 and Y0 are the time zero bacteria and amoeba. So this is Suav, who was doing the experiments, and Emmanuel, who was modeling it. And what you can see here is, and I know it's a little hard to see, but these are the different bacteria. This is the different amoeba, the different colors that you can see here. These are the different conditions. And what we have seen is that specific condition would increase the decline of the bacteria, while the increase of the and at the same time, the increase in the population of the amoeba. I know it's a little bit hard to see, but what I'm going to show you is kind of summarizing that for all bacteria and all amoeba, and it varied significantly between the bacteria and amoeba. I'm not going to go into that. It's more complex, but what we have seen is that under higher alpha and higher beta, we have an increase under very specific condition, which were increase in salinity and decrease in temperature, which kind of tells us that what we're seeing is that under high salinity, when bacteria are not feeling or are might be more vulnerable because they're susceptible to salinity, and maybe lower temperature where bacteria are not as motile, then the free-living amoeba can actually predate over the bacteria. So in conclusion, we kind of think that at low temperatures, high salinity, it increases the efficiency and the fitness of the amoeba predation, which we found to be very interesting. And basically, the overall kind of take-home message from this talk is that trophic interaction in soil have a really can have a really big influence on the contamination. So it could be pathogens that we're introducing into the soil, and it could be antibiotic resistance, and it could be or antibiotics that are introduced, or different types of contamination that we're introducing to the soil while we're irrigating with treated wastewater. So we're following that up. We couldn't have done it without these really amazing people coming from different places without funding. And thank you for listening.
Well, thank you very much. So any questions, comments? Yes.
Okay, so Osnat, I'm intrigued about something. It is known for years that one of the predation by protists and by amoeba is limiting the size of cyanobacterial mats. It's a very old concept. There's the alpha, and the correlation is basically almost the same. And it's also very well established that amoeba are like an amoeba histolica is predating on E. coli and salmonella. So in that sense, it's moving the cyanobacteria system where you know that the bacteria are regulated by amoeba to the soil. This is the main important step. And the question is, did in the many other systems that already studied it, did they see the correlation to high salinity? Because there were experiments like that in water.
I'm not familiar of salinity-mediated predation interaction studies. There are some studies that were done with temperature. Most of the studies that are of interaction are very old. So they're the 50s and 60s. So it's this maybe. So we haven't seen them. But basically, we haven't seen any studies that looked at salinity in soil. And I think that in soil it would be very different from freshwater. We also know that the protist community in soil is very, very different from the protist community in water. Very, very different type of amoeba. And so I don't think we can draw conclusions. And we are really trying to go forward with looking at amoeba that are found in soil. So it's both of these. But we haven't seen any studies that directly looked at salinity. We did see about temperature, and the results were very similar to what we've found. Camille.
Thank you for the talk, Osnat. You're talking and also using antibiotics in soil. Do you have any idea how stable these compounds are a long time?
No, we haven't tested that, but we are looking at the microbial community and the protist community just to see how it may affect the presence, how would the antibiotic would affect the presence of the community, because this is what's important to us, like looking at the effect on the community.
Well, thank you very much. So the next speaker will be myself. I'm sorry, yeah. So okay. I will talk to you today about family ties, what we call root social physiology within the Solanacea. I just like to thank all the people involved in this. So Dr. Omri Finkel, Dr. Milena Oliveira, who was a postdoc in my lab, Dr. Shika Verma, who is a postdoc in my lab, Ayn Yeko, who is starting her PhD. And I would like also to say something that I usually don't say. I would like to thank Professor Ariel Novoplansky, because all this is, the research started from his inspiration to me, and really, I really appreciate the window you gave me to go in. So root-root interactions. So as we heard today a lot, there is a detection mechanism that involves a capability of self, non-self recognition that was shown. So so a root has many roots, and this root somehow knows that this root belongs to me, to one individual, and this root knows that the red is belonging to the red, but the blue is the blue. So there is a mechanism which is a self, non-self mechanism, and we heard about it a little bit. And this is a doctor, already Professor Ina Meyer, who a long time ago came to do a postdoc in my lab. And when she came, we had these plants inside, and they died. So I didn't know what to do. So then I heard about Ariel's research. I said, let's do quickly an experiment that will have something. And then we grew plants in 3 pots. So these are pea plants. And this is so this is 3 pots here. So in each pot we have peas, one individual. So these are self in each pot itself. And here in these 3 pots, we split the roots where we have in each pot roots from 2 different individuals. So this complex is a non-self individual, and this complex is a self individual. And then this is how it looks. So we use the peas, and we measured the root respiration. And what we saw was very exciting for me, that when you are alone, you're respiring relatively low. But when you are with a neighbor, you are like stressed and go, you are 27% more respiration. So this is like being in an elevator alone or not alone. So you are nervous or not nervous. But this is respiration. This is really, and this was published, and we were very excited about that. But this was not sponsored, the research. So we left it alone, and we did many things since then. And then we received, together with Omri Finkel, this ISF grant, because we were all the time thinking, let's do. And we decided to look, and this is what I'll show you today, about the Solanacea family. So we looked at the family, what's happening in the family. So this is a cherry tomato, field tomato, and bell pepper. Yeah? So, and what we try to call it is that we have different degrees of relatedness. So when cherry is with another cherry, when it's alone, it's alone. When it's with another cherry, it's like with a brother or sister. When it's with a field tomato, it's like with a first cousin. When with a bell pepper, it's like a distant cousin. Okay? So this is degrees of relatedness. And actually, it came back now from new phytologists to revise, and they don't like all these terms. They say too much. So we will think how to fix it. So I'm not sure yet, but this is the thing. So this is so just to show you, CC is cherry with cherry, CF is cherry with field tomato, and CB is cherry with bell. Okay, so these are close related, and this is far related. So the setup is understood, and these are the three great scientists who did the research. So this is Aye and Milena and Shika. Milena left to the US. So what I'm showing here is all their work. So it's not, I didn't do anything. Yeah. So this is the first thing. So in order to do a lot of work in repetitions, we grow plants on paper. We call it rhizoslides. And this is how we did. And we had these 6 different treatments. So CC is cherry with cherry, like brother and sister together, yes? And this is, again, brother and sister, field tomato with field tomato, bell pepper with bell pepper. And then we had these 3 combinations. C and F is like having a cousin. C and B is having a very distant cousin. And FB, again, distant cousin. Okay, so this is and this is how it looks. And we look at the physiology of the shoots and look at the root dynamics, and we look at many things. So I won't show you all the things we see, but the first thing, so when you do a repetition of an experiment, you're always afraid, because repetitions are source for errors, right? Without repetitions, you won't have standard errors. So this was very nervous thing for me to do after we did 10 years after to do it, and I was so excited when we saw that cherry with cherry is respiring like this. When you put it a cousin, it's going more, and when you go it to the further cousin, so okay? So if you were someone you're close to, you're respiring a little bit, and this was true both for cherry and field and bell pepper. So for all of them, we saw this. If you are with someone close to you, you're respiring less, with something more distant, and I won't talk about the distance, okay? But it's more distant, okay? So you have more respiration. So this was really comforting, and really I was nervous that it won't happen. So then we look at the exudates. You talked a lot about exudates today, and we see that this is the root exudates, the carbon in the exudates. So we see that we didn't still analyze the different carbon compounds, but we see in total, when you are with a distant relative, the total carbon in the exudate decreases. So the exudate composition is different depending on your neighbor, and the distance and the degree of relatedness of your neighbor. And this we saw also for protein. So you have less protein in the exudates. When you are with a distant cherry with bell, you see you have less protein. So the composition of the exudates is different also in carbon and in protein, depending on the degree of relatedness with who you are. And then we look at a specific root length. So this is the area per weight. And we see, so when cherry is with cherry, it has low specific root length. But when it with more distant relatives, this specific root length increase. This is right, again, for bell pepper, not for bell pepper, for field tomato. But in bell pepper, we didn't see this at all, okay? And this, if you have this is like indicating thinner roots, okay? When a distant relative, you have more thinner roots, okay? That can be more functionable also in uptake. So the tentative conclusions are that respiration and specific root length increased as the degree of relatedness decreased, and exudates carbon and protein decreased as the degree of relatedness decreased too. So then we went to go and we had to we wanted to add the self, non-self, and to add salinity. And the idea for salinity was, okay, we have higher respiration, what does it do? So in my opinion, respiration gives you energy, and will it help the plants with the acclimation to the salinity? So we had these treatments. So we took out the field tomato, in order to make it more easy, because it's really a lot of work. And we used only cherry and bell. So, and either cherry alone, or cherry is cherry, bell, and cherry is bell. And in order that the people always say, okay, maybe they have more space, the roots. So when you are alone, you have half the space. So the space is the same. Okay, this is just to save you the next question after the talk. So again, we look respiration. We saw in cherry increases. Okay, and when you have salinity, it increases more. Yeah, the respiration, but this is known, okay? That respiration, salinity induces respiration because the plant needs energy. But please note that the increase here in percentages is much more significant than here. So there is something. In photosynthesis, this was very, very interesting. So photosynthesis, again, when you have a neighbor, cherry bell is increasing its photosynthesis as being alone, okay? And also under salinity, you see, under salinity, if you have a distant relative, you are doing better than alone. Okay? This you see here versus here. In bell pepper, it's completely the opposite. So cherry likes to be with his relatives. Bell pepper wants to be alone. Okay? He doesn't like his neighbors. And this is what we see here. This is photosynthesis and shoot dry weight. And but when we look at the root, this is root, specific root length or specific root area, so again, we see this increase both in bell and cherry. So when they are with a distant relative, their efficiency of their root is increasing, okay? So this is true for bell and under salinity, it's increasing more, okay? When you are with a neighbor. So the tentative conclusions here is that above ground carbon metabolism behaved oppositely in cherry, which is a social species, versus a bell, which is a loner. And salinity enhanced these behaviors. And neighbors' presence, I didn't show you a lot, but induced acclimation to salinity, mainly in cherry, also in bell pepper, but in cherry it's clear that if you have a neighbor, you are acclimated to a future stress. And then we have Dr. Shikha Verma, who is doing things that I don't know anything about, but she's in my lab. So she's doing metabolomics, transcriptomics, and data integration. So we're in the lab, we are a shovalomics lab, you know, so we only use the shovel, but so we bring doctors like Shikha Verma, and we took samples from the root, and we did analysis for metabolomics, also for tomato and also for bell pepper, and under salinity and under non-salinity. So T is tomato alone, which is cherry, yes, TT is cherry with cherry, TB is cherry with bell, and with salinity. So anyhow, what we see here is very complicated, so different metabolites. But what you see first, salinity is affecting, and a neighbor is affecting. Both have a very big effect on the response to the metabolic, how it looks, profile. And then when I compare the metabolic profile of tomato and bell together, so for under salinity and non-salinity, so first you see this is a control, or right? This is control, this is salinity, so they change. But what you can see here, so this is all the tomatoes, when tomato alone or tomato with tomato. Here again, tomato alone, tomato and tomato. And when tomato is with bell pepper, it's like behaving like bell pepper. So it's when you are next to bell pepper, you are having a costume and you are like a bell pepper in the metabolic profile. So root metabolome of cherry, this is what we see, is mimics the metabolome of bell when grown together. Okay? And when we did the transcriptome, we have this hierarchical clustering of the top 1,000 differentially expressed genes. Again, this is all new data, which I really don't understand a lot in. So this is tomato, this is tomato, tomato with bell, tomato with tomato, and this is salinity. So of course, again, we see salinity has an impact, but also the gene expression is completely different when you are, depending who is your neighbor. And we could go into it and look at which genes are related here to root development again, it's completely different. And we have so much data with that, I don't know what to do with it. So it will take so many years maybe not, but for me, it's really, so Shika is working very hard, and we have a lot of it. And so the next thing is everybody's telling me, okay, what you're doing on paper, pods, no, it's not worth anything, okay? So just to say aside, for me, working on osmo pods and paper is very important. It shows the potential, genetic potential of every species. Because also when you're in the field, so every field is different. So also paper is some kind of field, I'm pretty sure. So this is my opinion. I'm sorry, but some agronomists won't agree with me. But we decided, okay, let's go to the field. And this is an ongoing now experiment in the R&D you were 2 days ago. So what we did, we decided we are going to the field with cherry, tomato, and bell. And we're doing self, non-self. How we do self, non-self? Self, we just put in the ground like a half a meter of a screen between the two plants. So the self, non-self is only the below ground. And we have cherry, bell, and cherry bell, either self or non-self, okay? This is the experiment. And with salinity, without salinity, and you know it's not easy to do because I don't know if you know, in order to get fruit, you need to pollinate tomatoes, but tomatoes don't have a lot of nectar, and bell pepper do have nectar, so you put bees inside and they go to the bell pepper. But we looked for a cherry tomato cultivar that is a little bit more pollinates, self-pollination, and maybe has more nectar. And we didn't see it, the yield, because it's ongoing. But what is just now came last week is some results, and this is amazing. So cherry in the field alone, this is the black is the control, red is salinity. So the salinity is we know that it's not affecting a lot cherry tomato. They are more tolerant to salinity. So you can see here, the bell pepper, this is the plant height, are bigger. When cherry, this is cherry tomato, sorry. When the cherry is with the bell, it's significantly higher than when it's alone or with cherry, taller, yes, taller. And also, so this is the plant height, and this is in cherry, exactly the opposite. So here we again see cherry likes to be with friends, and bell pepper is a loner. And again, I think we also see the acclimation of the salinity. Let's see here. So we see, again, so this is exactly, somehow, this is the bell and this is the cherry. And you see the photosynthesis in bell decreases when you're with a neighbor. In cherry, you really don't have a big difference. So we had many effects, and we tried to put it in this model. I won't go over this. And to conclude, neighbors' presence and identity change plant responses and induce acclimation to salinity in cherry, and also in bell pepper, but less significant. Cherry and bell behaved oppositely when grown together, both on rhizoslide and in the field. Cherry is a social and bell is a loner. Thank you very much. I see my time. I'm okay. So.
Not only its own.
Yes, everything is, yeah. What do you mean? The neighbors, yes. No, we did. So what do you mean the neighbors?
There are 2 plants, right?
Yes. Both were analyzed, the cherry and the bell. And when I show the results, it's from the cherry or from the bell. When I saw cherry with bell, so it's the cherry. When I say cherry and bell, so that's the bell. And cherry, both are analyzed. Okay. But the question is yes, and not only that, we repeated it in a pot experiment where it's easier to do this. I think what you're meaning. Yes, Tamir.
Thank you for the talk. I was feeling about the bell pepper radical. But this seems like something very fundamental for competition in ecology. So why do you use a crop plant for this?
So the question is just because this is what we wrote in the proposal. Yes. This is the answer, and most of the people who were doing all these interactions, they didn't do it on crops. All the less knowledge on crops, and I don't know. Naftali is all the time saying to me, "You're working on plants you cannot eat." So here I'm working on plants that I can eat. So sometimes, but ecology is important. Yeah. Yeah, food security. Yes.
So when in the first experiment, with you call them brothers, what was the relatedness? Do you know the relatedness between the seeds that you used for the brothers?
So this was, I was asked also by Vered. The answer now, I don't know exactly. So the relatedness is cherry with cherry and cherry with field. I said, when I talked, I said, I don't know the exact relatedness. I will have it. I didn't do it, yes. So, okay? Yes, there are hybrids. Yeah, but this is the same. The fact is we see the differences, okay? This is, for me, it's enough, really. So if we take 5 degrees, yes. So I would like to really conclude my talk and go to my job as a coordinator and give our last speaker for the conference. And our last speaker from the conference is from Morocco, from the Maskir Institute, from the M6UP. And really, I'd just like to tell you, if you don't know, this is the first time we have Moroccans here. So 2 years ago, it was not possible at all. And this is amazing. I'm really, really, really so happy to have Dr. Aasfar Abderrahim. We had other speakers from Morocco who left already. I think they left you alone. So, okay. But we hope we'll host you good. So thank you very much.
Thank you, Shimon, for this nice introduction. Just to let you know, I'm not as funny as Shimon. Just support me. So, okay. Yep. Today I'm gonna share with you the results of one of our projects, which deals with atmospheric nitrogen fixation under different fertilization regimes. And just before starting, I would like to tell you a brief overview of our foundation and what we are actually doing. Masir Foundation is a non-profit association which is part of the M6P University. It aims to promote and develop technological poles in Morocco in the field of materials, nanomaterials, biotechnologies, microelectronics, and life sciences. So to move on to motivations, as you all know, nitrogen is one of the essential elements for plants. It constitutes around 1 to 5% of the total plant dry weight, and he is a major element in plant and building blocks such as amino acids, proteins, and nucleic acids. He plays also a major role in photosynthesis and plant energy. This makes it the most required element by plants and can be the limiting factor for plant productivity and proper crop development. And right now, to meet this increase in demand for food, notably in developing nations, farmers rely on the heavy use of nitrogen fertilizer, along with other fertilizers, of course. But up to 50% of the application of this conventional N-based fertilizer is subject to loss in the environment and the soil, which could substantially inflict economical and environmental issues such as greenhouse gases and soil acidification. And what you are seeing here is the Haber-Bosch process of manufacturing ammonia. This process is considered among the most energy-intensive process in the world. And this process contributes about half of the nitrogen inputs in agriculture, which means that about half of the nitrogen atoms in the body of an average person once passed through and participated in nitrogen to ammonia Haber-Bosch process. In this context, there is a growing need to consider new innovative approaches, less dependent on conventional fertilizers for more sustainable and smart agriculture. And one of these approaches is biological nitrogen fixation. It is a natural process for changing the atmospheric nitrogen into a soluble, non-toxic form directly used by plants. And there are several species all over the world that can do that using an enzyme called nitrogenase. We are interested in the non-symbiotic ones, which are living freely in the soil with no direct contact with plant cells, especially for crops who cannot establish this kind of relationships. For example, cereals. And besides BNF, this bacteria could also promote plant growth like by other things such phosphorus solubilization, fish hormones production, production of siderophore, polysaccharides, and enhancing nutrient uptake. So the aim of this work is to develop. The aim of this work is to take advantage of this natural process in order to develop, to reduce the chemical inputs, to improve the efficiency of the applied fertilizers, mainly phosphate, to improve the yield of the target crops, and also to develop new formulations using a local rock phosphate that we have at Jallat in Morocco. And to do that, we started a workflow. We followed the workflow that started with the sampling of the soils from different regions in Morocco, as you can see here. Then we brought them to the lab. We started the conventional isolation using a medium without nitrogen. Then once finished with the isolation and purification of strains, we passed them through a PCR amplification using several types of specific primers to the nitrogenous gene. And among almost 400 species, we found only 23 species that exhibited this gene. So we took these 23 strains and we started our screening. The first screening test was the ARA test, to quantify the capacity of this strain to fix nitrogen using gas chromatography. And the results showed to us that several species has this capacity at different rates. Then we moved on to phosphorus solubilization. We used two types of phosphorus here, rock phosphate from Morocco and the local TCP commercial one. Then we moved on to the quantification of andyl acetic acid oxide by considering an important phytohormone for plant growth, especially for the root parts. And we identified these 23 strains using the 16S sequence analysis, and we found several types of bacteria, but the main bacteria, the abundant species were Pseudomonas and Bacillus. And for the last test, we subjected these strains with plants in gnabiotic conditions using the MS medium without nitrogen. And the results showed to us that several species can really promote plant growth in this condition compared to the control. Now we finished our screening and we want to move on to test this strain with plants, but this time in combination with fertilizers. And to do so, we had to reduce the number of the strain and to choose only the potential isolates. And to do that, a two graphical projection by PCA was made where all the parameters were correlated between each other, and we found that plant parameters are highly correlated with nitrogen fixation, as you can see here, and phytohormone production for those two strains, so we took them. And we chose also the 63 strain based on her results on phosphorus solubilization. Now we want to move on to test this strain with plants and in combination with fertilizer. And before doing that, we had to do a viability test of these strains in combination with fertilizers and liquid medium. And the results showed to us that the combination with fertilizers did not impact the bacterial growth. In fact, they showed similar growth to the plants, similar growth to the normal growth. Some of them showed a lag phase before rejoining the normal growth. Some of them cannot tolerate high concentration of fertilizers. And when I say high concentration of fertilizers, I mean more than double that of what the plant actually needs. And we moved to our agronomic test here. We choose two types of phosphorus with 3 regimes of nitrogen, the full needs of plants, and half of that, and 0 nitrogen. And the results show to us that these strains can really promote plant growth. As you can see here, and some of strains, and this increase could be more than 50% for some strains, as you can see here. We also checked for PNN and elements in these strains, and we found that both of them follow the same pattern as here. Another important finding, as you can see here, this is the control who starts wilting just after 6 weeks of growing, while the other ones were growing normally. And as you can see here, this is the P content in plants. We say that this P content in plants increase is mainly due to the plant enhancing, the plant assimilation enhanced by bacteria. And this was for JSP fertilizer. Now we move to RP fertilizer. In general, the culture with the test, agronomic test with the RP was a little bit, RP little bit of slight of growth, and that maybe can be mainly due to the complex form which is in it. Yes, and here the 63 strain proved was that we had made a right choice by choosing it to use it with rock phosphate. Then this is all of this was at lab scale. Now we want to move on to the field or to the greenhouse. We know that in order to do that, we have to project our strains to put them back into the soil again. And for that, not me, my friends, did a lot of work on formulation, liquid formulation, powder formulation by atomization, and formulation with pH and rock phosphate. And according to the results, which shows we followed also their cell viability for like 8 months now and counting, and we chose this one because of its results and also because we want to make a good use of our rock phosphate. And then we launched another test in a greenhouse this time. This time we use 4 levels of nitrogen with 2 types of phosphorus, and the bacterial strain was used in the form of formulated and not formulated. And the results show to us that the results confirm to us the capacity of this strain to really promote plant growth. And that's it. So to conclude, the selected strain have significant potential for reducing the use of nitrogen chemical fertilizers while confirming the ability to be used with the complex sources of phosphorus. And also the yield obtained showed that these strains were able to reduce chemical nitrogen by up to 20%, 25% using GSP and rock phosphate. And also the formulation that we developed worked very well in this test. And right now we're trying to develop a low-cost medium for the mass production for these strains. And also we're trying to understand the mechanism of this increase and everything. And here you see my the lab team that work in this project. And here is my email if you have any other needed information. Thank you.
So thank you very much. Any questions, comments? Yes, Eddie.
Have data on how viable they are in the soil over time, if you have to keep reintroducing them or if they can stick?
Well, in the soil, no. We have a data just on the formulations, just in the lab. We kept them in the lab and we followed their cell viability in the lab. But in the soil, we did not check.
Any other questions, comments? So thank you very much for all our speakers. We conclude, we have lunch outside, so we have these cool lunch bags. And I hope we forgot to do with everybody a photograph of the conference, so I hope you stay. We'll try to do a photo, okay? Okay, thank you very much.